Hello, my name is Joseph Romano. I am a rising senior plant science student at the University of Delaware, and I spent this summer as an Envision scholar. During my time this summer, I worked under my mentor, Dr. Chi Mu, on a project related to better understanding the interactions between the growth of maize plants and a pathogen called Pythium. Pythium itself is a soil-borne fungus-like organism which is classified as an oomycete. This means that it has a filamentous body and spreads asexually via structures called zoospores. The pathogen can also spread sexually through a means known as sporangia and can cause massive economic losses in infected fields across a wide variety of crops. In most cases, symptoms include damping off and root and crown lesions, leading to rot. In fields across Delaware, Pythium is a massive issue on corn, with the strain Pythium graminicula being chief among causative agents. Ongoing experimentation seeks to understand the pathogen better, with the goal of understanding more about the genetic basis of the infection and breeding for resistance. My project seeks to aid in this study of the pathogen by trialing different growing methods for greenhouse experimentation to find ideal methods for application in future projects. In particular, we hope to gain insights into the best practices available to evaluate Pythium's direct impact on the root system as a whole. To begin with, my experiment has a variety of different variables at play. These include the maize genotype, disease status, and container used. In terms of genotype, equal numbers of both B73 genotype, which is a well-known and studied inbred reference variety, and either Will 900 or PHT22 were used. Both Will 900 and PHT22 are genotypes with moderate resistance according to previous work, so we hope to test them here. In regards to disease status, a control group was included with both Pythium graminicula and Pythium dosaticum as well. These strains were selected due to previous experimentation finding relatively consistent infection of the Pythium graminicula strain and a positive beneficial aspect of the Pythium dosaticum. Lastly, container type was altered with the goal of seeing if cone containers provided better access to roots during experimental takedown through limiting root binding, and as well to see if they saved space or led to more infection than the previously used square pots. In general, we hope to compare the root architecture of the two container types, as the root traits are important in rating Pythium infection. All told, the experiment includes 96 plants, 48 per genotype. As you can see here, the first step of the experiment included gathering all different materials. This included all the pots and trays for each group, the seeds, as well as the pro-mixed soil used as the media. The pathogen itself is cultured on media plates and then transferred to a bag with sand and cornmeal, allowing it to be spread evenly through the media, as you can see here. We have to be careful to approximately weigh the media to allow for equal distribution across each replication. Once our media is mixed, it is time to set up our trays for both the square pots and cones. After these are filled with dirt, we then put the seed in each tray, tag them, and then move them to a greenhouse bench where they'll be grown out. Plants are kept in trays which are filled with water to encourage the spread of the pathogen's asexual zoospores. A final watering is the last task for setup day before we wait for emergences. In general, first emergences can be noted after 5 days of planting. It is not extremely evident yet, but as the plants continue to grow, more symptoms will be noted. Daily tasks include watering the trays and recording data about emergences, with a lot of pictures being taken. Imaging was undergone before the plants underwent a full trait collection period. Taking professional pictures of the plants is extremely valuable for down-the-line analysis, especially in the case of comparing treatments and growing methods. At the end of the 21 days growing time, experimental takedown was underwent. This included taking each plant and measuring its final growth stage, root and shoot weights and lengths, and giving each set of roots a rating 1 to 10 based on lesions and quality. Once each of these traits were collected, roots were then taken to the lab for imaging. These images are to be later digested by a high-throughput phenotyping software called UD Envision, which can then generate even more traits that can help us define underlying interactions between the plant and the pathogen. All in all, my project serves to help better understand the methods by which we can study the pathogen Pythium in a controlled environment. By experimenting with different containers and genotypes, we can better understand not only the differences between genetic backgrounds of maize and their tolerance to Pythium, but also learn the best practices to use when studying the pathogen going forward. Thank you for watching.